Leading a startup team? Whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Caelum Biosciences, a Fortress Biotech company, is a clinical stage biotechnology company developing treatments for rare and life-threatening diseases. Its lead therapeutic in development is a novel antibody to treat patients with amyloid light chain amyloidosis, a rare disorder of the bone marrow that causes misfolded proteins to accumulate in tissue and organs throughout the body and cause damage. We spoke to Michael Spector, President and CEO of Caelum, about the company's business model, its lead therapeutic, and how the competitive landscape for a rare disease therapy may shape development strategies. Michael, thanks for joining us.
Thank you very much for having me.
We're going to talk about Caelum Biosciences, AL amyloidosis, and your experimental therapy to treat it. Before we do that, though, I'd like to talk about business models. Caelum is described as a Fortress Biotech company. Fortress created Caelum at the start of 2017. What is Fortress and how does it work?
So, Caelum Biosciences is a subsidiary of Fortress, as you've mentioned. Fortress is a parent company who seeks to identify great science happening throughout the world, whether it's at universities, smaller biotech companies, larger biotech companies, where that science is not able to progress in its current form for whatever reason. We have a business development team that tries to identify those opportunities, and we license those opportunities either for the purpose of initiating a new company, if it's a scientific area that a Fortress company is not already in, or to fit into an existing company that's already established.
Now, what's the benefits of this type of approach? What you able to do things more cost effectively, faster?
Sure. So I'll give you an example of how it worked with Caelum Biosciences. So we licensed Cal 101 from the Columbia University in January of 2017. The benefit to us is that we become a fully functioning biotech company at day one, because Fortress has staff in most of the major biotech disciplines. So we have a group that focuses on formulation and manufacturing. We have a preclinical group. We have a clinical operations group. And we also have the back-end functions of finance and human resources. So as the CEO of a new company, while I have to hire people who are specific to the category, at day one, we can hit the ground running and start moving as a proper biotech company without having to form, interview, and find all the proper people to support the company.
Well, the asset you licensed is for a potential treatment for AL amyloidosis. What is AL amyloidosis? How rare is it? How does it progress? And what treatment options exist today?
Sure. So AL amyloidosis is an amyloidosis that starts at the bone marrow. So our bone marrow produces plasma cells, and from there we produce proteins. In the AL amyloidosis patient, you get a misfolding of those proteins. Those misfolded proteins aggregate together and then deposit throughout the body. So in many cases, it's in the heart, the kidney, also the liver. It can deposit in the skin and throughout the system. We're most worried about the heart and the kidney, and that's because in a patient with cardiac amyloidosis, you end up having patients progressing to severe cardiac heart failure, and when they're diagnosed and properly diagnosed, a large portion of these patients have a high mortality within the first year. It's estimated that there are about 30 to 40,000 patients within the U.S. and Europe, and probably around 5 to 6,000 new patients a year in the U.S. and Europe. The difficulty with calculating the number of patients that exist, and we believe that there's an underdiagnosis in this area, is that many patients are misdiagnosed early, and they see 3 or 4 different doctors, oftentimes a cardiologist. This is almost like a needle in a haystack for a cardiologist. They may see only one of these patients every 3 or 4 years, if ever, given the rarity of the condition, and therefore they're not actively thinking about amyloidosis, and the patient isn't diagnosed. At some point, in many cases, the patient gets a biopsy, and it's determined that they have amyloid in their organ, and then they're sent typically to a myeloma specialist or an amyloidosis center. And the reason why often the same treaters of multiple myeloma are the people who also treat AL amyloidosis is that they're both plasma cell dyscrasias, and therefore the myeloma specialist is very comfortable treating with the same therapies that you would use for myeloma are also used for AL amyloidosis.
So how is AL amyloidosis treated today?
So the term that you'll often hear is plasma-directed therapies. Most frequently, those are chemotherapies. What is thought, the objective of treatment is to shut down plasma cell development, and therefore shutting down the protein cells that the proteins that misfold, stopping future amyloid from developing. What's different between the treatments and the promise of CAL-101, our lead antibody, is that we're seeking to demonstrate that CAL-101 works at the site of the amyloid. The current hypothesis on how CAL-101 works is that it binds to the existing amyloid within an organ, and it stimulates our own bodies to have an immune response and break down the amyloid. So unlike the chemotherapies and plasma-directed therapies that work at the source of the condition, they are less effective at breaking up the existing amyloid. We're seeking to develop a treatment that works at the source of the amyloid and work in a complementary fashion with the plasma-directed therapies.
What was the process of taking the asset and deciding to create a subsidiary company around it while you were at Fortress? What was your role in that process? Did you identify the therapeutic? Did you work on preclinical development at all?
Yeah, so when we identified the opportunity, it was already in a phase 1 trial at Columbia University. To take a step back, CAL-101, which you'll also hear referred to as 111F4, that was the original name of the compound, emanated from the University of Tennessee. And the University of Tennessee actually really initiated the whole development process of treatments to attack the amyloid, and it started about 20 years ago. The original inventor, Alan Solomon, of the concept of directly treating the amyloid deposits, retired and transferred the program to Columbia University. And Dr. Suzanne Lench, who is the principal investigator on the phase 1 trial. I had met Dr. Lench probably in the first half of 2016, and I got to know her well and understand the work that she was doing with CAL-101, at the time 111F4. There was some very, very preliminary phase 1A data that had already been presented at the ASH meeting of 2010, which is the biggest hematology meeting in the United States. We were very excited about what we saw from the single dose portion of phase 1. And she was planning to present the full phase or preliminary phase 1A data. Sorry, I'm going to start over. Dr. Lench was planning to present the phase 1A results as well as preliminary phase 1B results at the ASH meeting in 2016. And so we had signed an agreement with Columbia University, an option agreement giving us the rights prior to her presentation in 2016 because we saw the potential of this as a future treatment, and we wanted to hit the ground running in 2017 after the ASH meeting. We exercised our option in early 2017 and have been working hand in hand with Dr. Lynch since to try and get this into further development. And our plans are to initiate phase 3 clinical trials in the second half of 2018.
And this is actually an antibody.
It is. It's a monoclonal antibody that binds. So you'll see often that the term is a conformational epitope. When Cael-101 doesn't bind to the light chain, which is the plasma protein, doesn't bind to it when it's in its normal form. But when you see the protein misfold, there's a new epitope that's formed, and Cael-101 binds to that new epitope. And when that happens, we believe what occurs is that the body has an immune response stimulated, and neutrophils and macrophages are called to the site of the amyloid in the presence of that bound antibody.
At this year's ASH meeting in Atlanta, you presented data on phase 1 trial. What did the data show?
Sure. So the phase 1 data was primarily a safety study first, in that we escalated the dose to very low doses up to 500 milligrams per meter squared. And in the phase 1A study, it was a single dose, and in the phase 1B portion, it was 4 weekly doses with the patients measured at 12 weeks. One of the most exciting pieces of data that came out of the phase 1 study was that patients saw an organ response defined by a 30% reduction in a biomarker called NT-proBNP. And we saw a 21-day time to cardiac response. Given that many of these patients have a high mortality early, the sooner that we can start to improve the functioning of the heart, the better. So we were very pleased to see a rapid action of the antibody. We also, given the short duration of treatment, up to 1 month, we saw a cardiac best response rate of 67%, which means that 67% of the patients had hit that response rate of a 30% reduction in NT-proBNP. And when measuring all of the organs, we saw overall a 63% of patients responding to treatment. We saw no dose-limiting toxicities, so it gave us a very good sense that we can progress this to further clinical trials, and certainly off of the back of the phase 1 results, we feel very confident that we can continue to move the program through clinical trials.
And is there a clear sense of what you'll be using as endpoints in the later studies?
Right, so it's still under evaluation. Previous antibodies in development have looked at both the biomarker NT-proBNP and then combined endpoint of hospitalization and mortality. We're evaluating whether we'll follow that path, but most certainly we will look at mortality as an endpoint, because the most important thing about this treatment, Cael-101 treatment, is that we're able to extend people's lives. And so there's a correlation between the biomarker and mortality. And so we think showing an early difference in the biomarker is important, but ultimately we think it's going to be most important to demonstrate an improvement in survival.
You're not alone in seeking to develop an antibody to treat AL amyloidosis. Prothena has a late-stage antibody in development. Any sense how your therapeutic might compare? Are you seeking to do the same thing? Is it a different mechanism of action?
So both are antibodies that bind to the existing amyloid deposits. At least that's what we understand. The antibodies were developed differently in that Cael-101, the Caelum antibody, was designed specifically to bind to the kappa light chain, specifically the light chain fibrils that are the causative fibrils of the amyloid deposits. My understanding about the Prothena antibody is that it was being developed for another amyloid type and had some binding against light chains. I can't speak in detail about the protein antibody because I don't know their data as well, but what I can tell you about CAL-101 that was very important in early preclinical trials was that the light chains are heterogeneic, which means that there are two major subtypes, kappa and lambda, and then there's isotypes underneath that. What's important is that for these antibodies to work is that you have to have binding across a breadth of the different subtypes that exist in patients. Early on, there was a study where human amyloid was taken with patients' consent before they passed away, and the investigators were able to remove the amyloid and test it in animal models. And what we've been able to show is that against both high-level subtypes, kappa and lambda, that we've seen that CAL-101 is able to completely dissolve human amyloid in animal models. Again, I don't know the breadth of the subtypes, but we think that's an important component for CAL-101. Certainly, I think when you look at the time to cardiac response as well, we think that a lot of the rationale behind the rapid response time in phase 1 has to do with high binding affinity across the heterogeneous light chains, therefore stimulating an outcome in a breadth of patient population. I also think it's important to note the cardiac response rate of 67%, given the 4 weeks of dosing, we think is a unprecedented achievement in this class.
Given that there's another drug in development that's also an antibody, and you're dealing with a rare disease with small patient populations, how does that complicate or shape your development strategy?
So ultimately, the most important thing is demonstrating that you work in the population, and if we believe that the binding across a multitude of the subtypes of kappa and lambda and their isotypes, ultimately that's going to be very critical, and it's going to lead to outcomes that are important for patients, both in speed to response and mortality levels. I think if there are real differences that are shown in clinical trials, ultimately that's going to be the most important factor of how people will decide which antibody to use. If one is working faster to a higher degree against a broader patient base, then I think that'll become the lead opportunity. And we feel very good about our profile coming out of phase 1. Ultimately, that's going to be the deciding factor. But we have learned certainly from Prothena's experience, and we want to understand patient types that will likely respond. We're assessing a similar population that Prothena has studied, but we're also looking at other populations that had not been included in the Prothena study as well. One of the patient types that can most benefit, and we want to understand whether an antibody can be effective, is the sickest population. So you have the NT-proBNP level that I've mentioned, which is a biomarker that's correlated with survival. Some patients have an elevated NT-proBNP, and patients are really deemed to be very, very sick when they start to surpass 5,000 and 6,000 in terms of their level of NT-proBNP. At that point, there's a patient population where mortality is very high, and it's uncertain whether a treatment can have an effect. And certainly we want to study that population because those are the patients that die the quickest and are most in need of treatment. So we're exploring populations beyond what Prothena has studied to see whether our antibody can have an impact.
And is there a plan for the company beyond 101? Is this company built to develop this one asset, or do you have a broader pipeline you expect to bring on?
Sure, I can't speak in detail because there's active discussions about additional opportunities. We certainly expect to be in the hematology field, myeloma field, and amyloidosis field, also potentially protein misfolding more broadly. You may be aware that there's other types of protein misfolding diseases. There's been a lot in the news about TTR amyloidosis based on success of compounds and treatments in those areas. And And so we're certainly exploring protein misfolding as well. But we expect to continue development and are actively looking at opportunities to fill out a portfolio in both AL amyloidosis, multiple myeloma, and protein misfolding conditions.
Michael Spector, President and Chief Executive Officer of Caelum. Michael, thanks so much for your time today.
Thank you very much.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.